Thank you very much for inviting me to this very nice symposium, which is actually a mixture of bioinformatics and chemoinformatics. But if you see in the title of the symposium, bioinformatics stands first. It's bioinformatics and chemoinformatics. So why is that? Because before you do some search for a drug, you have to find a drug target. Here, my title is like Listen in Symphony of Epigenomics to find drug targets. And why I'm talking about epigenomics? Actually, if you consider the cell, then the genes which are expressed in the nucleus of that cells, we can measure expression by so-called transcriptomics. They are controlled by a variety of proteins in the nucleus, which are called transcription factors. And the epigenomics is everything which is done with genome on the level of accessibility of that genome to transcription factors is actually anything which controls gene regulation. If we talk about genome, it's actually mainly we are talking about what to be expressed, what are proteins, how they are coded. But when we are talking about when and how six proteins has to be expressed, we should talk about epigenomics. So, and then how to use this epigenomic knowledge to find drug targets. So our approach is basically to, once we know which regions of genome are accessible for transcription factors, and they start binding to those regions, then we can reconstruct the consequence of events in the cell, and then by reverse engineering we can actually come up with targets. We call them master regulators. Through this cascade, they can regulate activity of the gene, and epigenomics is actually a key of that process. And then in terms of epigenomic, there are many different ways of how to measure it and how to detect it, and especially on the level of binding of transcription factors. The main method which is used there is so-called ChIP-seq. And on this slide, you see a picture from how this ChIP-seq is done. Basically, it's done with antibodies to that transcription factor. And through this antibody, we know where the transcription factor is actually binding by further sequencing and then mapping those reads of the sequence to a genome. But important to see is that the result of those experiments looks like that. So basically, we see a genome, and we take a transcription factor or a more broader element, which is before the transcription factors, or maybe after, we don't know yet, happening. So like modification of nucleosomes. So then you see so-called peaks. So you see like a little profile of some signal going on through the genome, saying that, okay, this factor, this protein is binding somewhere, but then there is a peak which shows you here where it's binding most. And of course, many attempts are done to detect those peaks using bioinformatics tools. And that's the first which attracts our attention. Look at the peaks, because you see them, they are there, and they may be the most important ones. But that's something what I want to actually discuss. Basically, those peaks may be not really the most important parts. But I would like to do it on the example of one important transcription factor, which is called RUNX1. And there is a controversy of that factor because it was discovered many years ago as a main transcription factor involved in lymphomas, in different leukemias, I must say. And it's even name of that, another synonym of that factor, IML, it stands for the leukemia. But in recent time, people found out that this factor plays a big role in other types of cancers. And the controversy of that is that sometimes you see that this gene is deleted, which leads to the cancer. Sometimes you see that this gene actually expressed highly, and that also leads to the cancer, or leads to actually to metastasis in that cancer. So that's why it seems this mode of action, how this factor are binding to DNA, is really to understand that we should actually understand how it works. And to analyze that data, we took data from Gene Expression Omnibus, where in 2019, the group of authors, did ChIP-seq experiment with RUNX1 transcription factor in a particular cell line. And the same people actually did also transcriptomics experiment. So they measured expression, how expression is changed of the genes when we actually knock out RUNX gene. And then they also see where the RUNX1 is binding to genome. So we actually took this data and we compared it. And at the right side, you see a simple diagram which shows basically upregulated genes, more than 300, downregulated genes, more than actually 600. And here you see those genes which are located near the biggest peaks. So those peaks of ChIP-seq where the factor binding strongly. And you see the very little overlap. It's only 7 genes which are upregulated, and they have this strong peak in the front of it. And by the way, this RUNX1 gene actually has 13 these strong peaks inside this gene, but this gene is not upregulated by itself. So there is something strange going on there. So what actually doing those strong peaks, and whether they really involved in regulation? And to understand that, we apply our tools which are stored in our TransFact database, which we're working on for many years, where we actually collect information about binding sites for a particular factor from various experiments. Then we build a model which is called a motif or positional weight matrix, and then we apply this model to find signals, to find motifs, binding sites, transcription factor binding sites in any region of genome of our interest. For instance, we can do it in the peaks, in these strong peaks, which we are considering here, for which were found by direct experiment. RUNX1 was binding to that place very strongly. And that's a result of that search. Basically, here, each line here, or each bar, represent one peak. So it's peak 24, for instance. This is 24, this is peak 182, so they just numbered. So they are somewhere in genome. And then these little arrows here, red ones, are actually predictive sites for the RUNX transcription factor. So it's just called here AML1. So as I said, it's synonym. So, and what you see here, these peaks, which are strongest peak in genome, they're simply full of those sites. So, of course, when you see so many sites, when factor sees so many sites in genome in one place, he doesn't resist and he just binds to it, like a bee to the honey. If you look at this places more in zoom, so as you can see here, this peak is located actually somewhere in genome in between 2 genes, and very far from any gene which is upregulated or downregulated. It's the strongest peak, and it's full of those binding sites. You see them just repeated there one after another, and this is like very strong binding sites for this factor. So what are they doing there? We don't know, but obviously we know something. we have to understand something, how this actually regulation of genes is organized. So we have to know this regulatory code, or we can rephrase it, epigenetic code. So we know a lot about genetic code, but we have to know about epigenetic code. So how these signals, these motifs in the genome, they control activity of the genes. So by just direct measuring where they're binding, it seems it's not enough. So it's like we have some text which we don't know the language of, and only a few part of it we understand, like which code for the gene, but the instruction what to do with this person here, where to go, when, under which conditions, it's encoding somewhere in this place in the text, but we don't know how. But the question is it the text actually? So if you consider the genetic code and how it was discovered, and I presented here one picture of the genetic code, which with some question marks here, and that's special for you because that's a genetic code which was discovered by a bioinformatics effort of my Professor Ratner, actually 2 years before it was in fact discovered by a big team in the United States. but it was fully theoretically predicted. Just a side remark. But actually, the major thing which was found out in the genetic code, and it was the fundamental principle of a genetic code, is that codons, when they encode the gene, they don't overlap. So they go one after another, like in a text. And the key was, that's how the codons put one in another, like in a normal language, like in English or in other languages, you have words and they go one after another, and then they don't overlap the words, but then you have the whole phrase. And that concept actually dominate in many places in molecular biology, and unfortunately also in gene regulation. Till now, we often believe that the promoters, they actually controlled in the same way. So you have some text in front of transcription start site, some nucleotide sequence, and there are some words which are coming one after another, and they give an instruction to this gene to upregulate or to work or not to work. But in reality, if you consider one gene, and you consider those motifs which are there in the regulatory region, in promoter region of that gene. So, for instance, here the gene, which is called MFAP, here the first exon, it goes actually on a reverse direction from right to left. So you see that there are many motifs there, but then when you start searching for them more and more, you see this picture. So basically, everything is predicted by many times by various types of motifs. So it's not a consequent text of motifs, it's something different. And here, so we saw that it's something really organized, like a parking place in Germany, but it seems it's something like a park place in Italy, where all these transcription factors just try to find opportunity to bind to their parking place. So, and that's indeed the case. We know that in different cell types, the same binding place, actually the same parking place for a factor, can be occupied by completely different factors like NFAT, NF-kappa B, CEBP. They bind to the same place, or even receptor VDR actually binding to the same place in promoter. So they can bind to the same place, which means the words in regulatory code are overlapping. And that's you see also in the real case. So this is a ChIP-seq, actually massive ChIP-seq experiment on one cell line, where this is like 300 different transcription factors, and this is one region of enhancer, which is occupied by multiple factors. Each line here is one factor, and they occupy in that place and very nearby, and they regulate somehow this gene. And the analogy what I see here is like in symphony. So when you have each factor like a different instrument, and they are playing all together in a symphony, and then actually even you can compare the presentation I showed with multiple sides in different lines. It's like when you have a score of a symphony, when you have one line for each instrument and they go in parallel. Or even better is actually analogy is this charm, Charmanka, when basically you have one line, and each line here is like one transcription factor, and it goes through the genome, some kind of machinery, and then at each time point you get several of the factors in near place, and they give an accord. They give actually the music. But if you listen just to one factor, you don't see the music. And here we see like a paradigm shift when basically genetic code was discovered after George Gamow said, forget about biochemistry. All these nucleotides just letter, and it is a text, and we have to find the language of that text. So here we can say, forget about letters. It's not a text, it's a music, and it goes in parallel. And that's maybe the main principle of genetic code. And what are these huge peaks? This is not in music. It's like a big drum, which we listen and we hear it the first place, and we see it's the most important thing, but it's just a drum. It's simply too loud. And you have to really tune back and see your music. And what happened, how this accord of the music is happening there when several transcription factors, which they are binding nearly to the same place. So they are actually binding to very closely localized motifs, and they interact to each other, and they by interacting they made this accord which sounds good. So basically, which creates this this kick to initiate transcription of that gene. And sometimes those sites are very close together. For instance, this is an example published in this paper where this RUNX1 transcription factor binding sites, which is underlined here, and this is a picture of DNA, and here this is protein, and that's another factor. It's called ETS1, which is bind on another side of the DNA and nearly to the same place, and they bind at the same time. And when they bind, actually, it showed that RNX is able to interact to ETS1 and make a kick to it so that it activates ETS1 and it activates altogether the expression. In each case, it goes in different ways. And bioinformatically, we found we created a genetic algorithm which tries to find this combination of sites in this multiple prediction of sites. I was talking about that many times, but this genetic algorithm actually is able to find which composition of which transcription factor binding sites is actually playing the music in that particular experiment. And in this particular experiment about RUNX knockouts, we analyzed it with our tool and found that this combination of motifs, which include actually RUNX2, and AML, these are all synonyms or similar motifs in our library, they have to be accompanied with other transcription factor to play that music. It's not just one factor is not enough. Then you hear just the drum. And also in the downregulated genes, you see a different picture, but again, you see multiple time RUNX here and here, actually, which tells you maybe something about this controversy. So the RUNX in different contexts can be either promoting activation of genes or repression of genes. And then depending on the context, as it showed, that can be leading to activation of cancer or repression of cancer. And actually, when we look more in detail in one of the genes, which is upregulated, for instance, this one, you see here a colorful peak. It's not just one color peak. That here you see the accord, how the music is sound. So these are including many factors together. When you zoom this peak, you see how colorful is the peak, how many factors are involved in regulating this activity of that gene. And that particular gene is actually very important. So it's a co-activator of the TGF-beta signaling. It's known to be involved in cancer, and it's actually associated with various cancer, including the breast cancer, which was a special focus of that cell line, which we're analyzing. And also it's known that when promoter of that gene is hypermethylated, it's correlated with breast neoplasm. So it's definitely clear that epigenetic of that gene, which we now trying to find more in details, plays a major role in also in that gene. And also, you know, we check, all known ChIP-seq experiment, and we see that there are a lot of various RUNX1 or 2 binding sites predicted by various ChIP-seq experiments in the vicinity of transcription start site of that gene. So as I described, those transcription factors like these happy guys here, they may interact with each other and bind to this one place in the genome and activate or not activate the gene. And that's a very fuzzy process. So they can come in one condition, but they go in another condition. And it's like it's shown now that it's worked like a phase separation. So when you have those factors like oil, and many factors can interact with each other, and they organize in such droplets in genome, and they regulate the genes. So in the last few minutes, I would say, okay, we found the factors, and now we want to go further, as you've seen in the first slide. We want to go to find the master regulators, and we want to find actually finally the drugs. And for that, we have a tool, we call it a genome enhancer, which can do it practically automatically. You put their transcriptomics and epigenomics together, press a big red button, and then it actually creates, it goes through many processes. It analyzes upregulated, downregulated genes. Finally, it finds the most important master regulators. It's even put them in the title of that report. And it does many classical bioinformatics in between. By the way, it's also found, for instance, that these genes which are upregulated and associated with much lower peaks, not those big peaks, but really it starts working with the low peaks, and those peaks which can be detected in promoters of those genes, they indeed, in this particular case, involved in DNA replication in processes related to cancer. But finally, this process creates the master regulatory network. So you see on that picture here below are the genes which are controlled by the transcription factors, which are shown here in blue. And they, in turn, are controlled by a big signal transduction network. And at the top of that network, we see those master regulators, which are potential drug targets. So now we had the point of finding the drug targets, but as you see, the most important part of that network is actually this epigenomics, and actually to understand how those genes are regulated by various factors in that particular condition. And that's the most flexible part. Actually, this signal transduction part is more or less standard. It can vary, of course, but it can be rewired. But most flexible is this low part. And that's why we call it actually working pathways in some of our publication, because this, like the lowest part of that huge colossus, these legs of that colossus of signal transduction is the most flexible one. So the epigenomic actually controls which gene has to be up or down regulated. Finally, our program actually is able to predict also drugs, which can work with those, interact with those master regulators. And of course, the first it does the typical drugs, so classical drugs, and it found actually, as it should, the classical drugs for breast cancer based on those targets which we found and on the knowledge which targets are targeted by this drug. But actually, most important for us in this context of that symposium is whether when we found those drug targets, can we actually do the drug screening using some programs from chemoinformatics, for instance, PAS. And again, as we talked today a lot about screening in this symposium, we did also our attempts to converge this PAS output into a screening score. When we using this, we call it a target activity score, we actually screening not the whole variety of genes in genome, but only those ones which we found in our master regulator search. We actually do the screening in this among these master regulators only, and then actually when we see when we see the predict the PA or using PAS, we construct a certain screening measure which tells us, which drug is actually most interesting in the context of that master regulator set. And for instance, this is a result of such screening, and it gives us, for instance, busulfan as one of the top candidate because actually it using PAS, using prediction of activity, we found that it predicts very high PA for those targets which we predicted as master regulators, among them also this gene we were considering just in a moment. And busulfan is known and very old drug for treating leukemia, and actually it's also used in already many years for treating various cancers, although it's pretty toxic, I must say. That's why nowadays it's actually substituted by more modern drugs. But as you see, the prediction was done completely unbiasedly, and this drug pops up. So that's in knowledge about busulfan, which is known in clinical trials for many neoplasms. But we have actually a bigger list in our screening. So busulfan is just on the place 2, but there are other molecules which were predicted by such a screening, which may be interesting in the context of the cancers which have a RUN-X mechanism behind. Okay, so when we talk about this regulatory code, I just want to finish. So as I said, we don't know what is written here in this text, but by trying to reconstruct this text by typical, text reconstruction with the concept that our words should not overlap, we may not find any meaning of that. But if you take a concept that words can overlap, then you can read it. For instance, you can read here, hello, Alexander, or you can read goodbye, Alexander. Completely different things, like in our example, when RUN-X actually either can activate the cancer or inhibit it. Depending on the context, you can read the same message either actively or positively, or you can read something completely else, like run Alexander fast, and you see that some letters are reused, but the combination of that, the accord, which is played in that symphony, is different. So actually, with that, I want to finish. And basically, this is our team, and this is our funding. And thank you very much for your attention.
So thank you, Alexander, for a very nice and musical presentation. And we have 3 minutes for question, and there are, I see already 2 questions on the chat. So the first one, why do we need so many transcription factor? Is there some kind of redundancy? Do you think we could safely block some of the redundant factor without influencing others?
Yeah, very, very good point. Actually, I think in evolution, it was going rather in increasing of number of transcription factors playing in the cells. And that's very interesting process. Actually, we see it also factually. So if you consider more simple organisms with a more complex multicellular organism, and just simply see what is the percentage of regulatory genes, it's increasing. And many of them are actually transcription factors. And the reason is that these multicellular organisms, they have to face with much more different conditions inside and also outside, and have to reply. Having the same amount of genes as the simple organism, I'm simplifying a little bit, you have to react, those genes have to react to much more different conditions. And that's why you have to encode in the same place, basically, much more messages. And how to encode it, you have to increase number of transcription factors. So I hope I answered.
Thank you. And 1 more minute and 1 more question. As an amateur musician, I was interested if there were different time signature in which genes are expressed. I'm not talking about expression pattern necessarily. Is expression rhythmically accurate most of the time?
Okay, I guess I will answer that in the chat. Thank you very much.
Okay.
You have 1 minute.
No, it's okay. You can still answer. You have 1 more minute.
Yeah, okay, good. So basically, well, it's a complex question. Let me think a little bit and I answer in chat.
Okay, thank you very much.
Okay.
And so we are now up to the last talk, which will be given by Yang Chen from Japan. So please, the floor is yours, and share your presentation.
Yeah, but I don't see her among the participants, unfortunately.
Shall we wait a minute?
Let's wait a little. Yes, sure. Maybe she is coming, but not yet.
Okay. So I guess in the meantime, if anyone has any more question to any of the speakers.
Yes.
Well, I can answer this last question maybe.
Yeah, please.
So it comes seem to be from a musician. I'm an amateur musician, was interested if there are any different time signatures in which genes were expressed. And I'm not talking about expression patterns necessarily, it's expression in rhythmically accurate most of the time. So he's talking about rhythmuses. And we know there are rhythms in many rhythms in the life, for instance, the life of a cell. For instance, it has to divide rhythmically. And definitely when there are gene regulation, which has to be actually regulated in a rhythmic way, there is a special mechanism which makes such a rhythm. But at the end, these are controlled by a special complex of transcription factors, which comes to that protein, to the promoter of that gene in a very particular time point. So there are sometimes rhythmical expression regulation. That's true.